Okay, so in terms of gene regulation, we had seen that can happen at different levels. Same is the case with the eukaryotes as well. Just some of these steps, they are relatively a little differently regulated in terms of eukaryotes. And again, this goes back to the same fact that the complexity in the eukaryotic cell is little higher. So for example, at the whole genome level, so the first four, these are specific to eukaryotes. These last four, these are same like prokaryotes, what we had seen. So that is in case of eukaryote, genome level specific regulation also happens because of the folding of the DNA molecule around the histone, right? So histones, they do like by process of methylation or acetylation and so on, they cannot allow the DNA to unwound, and because of that, they can like switch off the particular gene. So that is one of the method. Also, the number of genes that will decide the amount of RNA and protein synthesis. So ultimately, how many genes they are being exposed and how many of them are forming RNA and protein, that can be decided at a genome level. Transcription of RNA, the effect of histones to keep gene inactive, like allowing whether the what we call as switching on or switching, keeping it, the gene, particular gene, keeping it off, that can be done with the help of methylation of cytosine. That is very specific example, or it is also termed as gene silencing. This is one of the very common phenomena that happens in eukaryote, that if a particular gene is not required at a particular given time point in the cell physiology, then that gene is usually silenced by the process of methylation of cytosine. And that is just one example. Otherwise, there are several modifications to these nucleotides which can also silence the gene, that is switching off the gene. Post-transcriptional level, so again the same, like at the processing of the primary transcript to make it a functional mRNA molecule at that post-transcriptional level. So there are these ribozymes which acts as riboswitches, keeping the either DNA, sorry, not DNA, RNA active to inactive RNA. So these ribozymes, they will specifically add, let's say, poly A tail, and that is done by one particular enzyme. So if that gene is kind of blocked, or if that particular enzyme is not functional, which we call as ribozyme, then obviously that gene will not, or the mRNA will not get activated. So ultimately, that way the gene can be regulated in eukaryote. And the transport of RNA out of the nucleus. Now in prokaryote, this was not much of a question, right? Because most of the things, they are in within the cell without any nuclear membrane. So if the RNA molecule fails to undergo changes after transcription, then that RNA cannot go outside the nucleus, because the specific requirement of the mRNA structure also involves, apart from its own function, also involves the transport of mRNA from nucleus to cytoplasm. So if that is also regulated, the particular gene regulation can also be achieved. So these are the four specific ones in eukaryotic. Rest four, they are common, like how we had seen. So degradation of mature mRNA or mRNA stability, that can happen with the enzyme RNase. Translation level of mRNA, again this can be controlled by those various initiation factors and so on. All of those ribosomal or pre-initiation complex and ribosomal assembly, all of they can control the gene regulation. Post-translational level, again at the processing of the proteins, whatever modifications are required in the protein, that can also control the gene regulation. Proteins can be kept at an inert or inactive stage by not allowing them to modify. So that's also one type of gene regulation, rather the regulation of the gene expression. degradation of the protein products. So there are several enzymes which can break down the protein that can also be secreted. So again, that protein action will be reduced. So ultimately gene expression will be there, but the action of that gene, whatever is responsible for, will be reduced. So these are the different levels of gene regulation in eukaryotics. Any questions here? Anyone? Okay, so now there are few other basic types of genes that we need to be aware of, because some of these terminologies they keep coming every now and then. So these are basically the type of genes based on the function and activity, right? So for example, we had seen something called as constitutive genes earlier, which are also called as housekeeping gene. Now we know the purpose of housekeeping is to keep everything in order. Like when we talk of housekeeping as a word in general, that is keeping everything in order at our house. So these constitutive genes or housekeeping genes, these are functional genes at all the time in all type of cells and approximately at constant level, right? And these are needed for basic cellular metabolism. Like I had given you an example of glycolysis enzymes. Their expression is not regulated because they need to be expressed at all the time. So that is when these are called as housekeeping or constitutive genes. There is something called as luxury genes or non-constitutive genes. These remain inactive for most of the time and are switched on only in certain conditions. For example, again the special genes in liver and kidney, right? In case of liver, if a particular product has to be degraded in the sense metabolized, or in the kidney certain substance has to be like activated or like thrown out of the body, especially in terms of those ATPase molecules where sodium potassium ATPase channel will exchange the sodium and potassium. So there are several such situations where there are special genes acting to create a special protein. Special protein as in it is required at only special times and not every now and then. So these kind of genes, they are called as non-constitutive or luxury genes, which is exactly opposite of the constitutive gene, which are always required. Non-constitutive, they are required in certain conditions. Then there is something called as uninterrupted genes or continuous genes or collinear genes. Now these are specifically called so because we know a particular sequence in the gene, which is codon, will translate to exactly same sequence of amino acid. And this we had seen while we were looking at the term collinearity, right? So whatever is the sequence of the DNA will be the same sequence in the protein, and that is why they are called as uninterrupted genes or continuous genes or collinear genes. Now again, in contrary to this or opposite to this is something called as interrupted genes or split genes. Now this is the one of the very interesting phenomena that we had not looked at so far. So the message coded in the interrupted gene, that is in the exon, is not continuous and is split into several distinct coding units, which is separated by the non-coding DNA, that is the introns. This fact we know already. So there in the DNA sequence, there is exon, intron. So two exons, they are separated by the introns. And in the process, while when the mRNA is synthesized, these introns are removed and then exons are rejoined in RNA splicing. Now, what happens specifically in these cases is RNA splicing is one of the events which can lead to differential protein. What do I mean by that? Is let's say a particular amino acid is like 1,000 amino acid long. So in such cases, the same protein variant can exist into, let's say, approximately 700 or 500 amino acid long. And all three different proteins can exist in a one particular cell at one time point, or they can express at they can be present at different time points, and all three of them, they can have different function at again the same time point. So there are several such proteins which are known within the body or within the cell, which has different lengths of amino acid sequence, and this specifically goes to the fact that how the RNA splicing has happened. So let me just show you one example here. RNA splicing, because this will be interesting to understand. So little bigger, this. Okay, I need a better diagram and simpler one as well.
Sir,
Yes.
so prokaryotes have uninterrupted genes, while eukaryotes have interrupted genes.
Sorry, prokaryotes are?
They have uninterrupted genes, while eukaryotes have interrupted genes.
Eukaryotes have not always. Eukaryotes, they do have uninterrupted genes. So uninterrupted and interrupted genes. So let me show you here. So for example, this is the normal process of RNA splicing. So what happens is, this is the DNA fragment, mRNA, there will be exons and there will be introns. Introns are this thin line and exons are these blocks, right? So during the translation process, what we get is mRNA, which does not code for introns at all, and that part is removed. Now, even while this splicing happens, you remember we had seen that overloop kind of thing happening in the RNA splicing, and that overloop is kind of chopped off, and these exons are joined by the ligase enzyme, DNA ligase. So that was one of the process we had seen in the RNA splicing. Now, in this process, what happens is, let's say a part of exon also gets removed, part or complete exon itself. Let's say this green one directly joins to the exon 3. That means whole of the exon 2 is gone, right? And that is a hypothetical example. It can be any of the exons or it can be multiple of the exons that can get removed during the process of RNA splicing. And this is at times very much essential, because those shorter fragments, so obviously the mRNA will be shorter than the regular length, and then the protein will also be of the shorter length than the regular protein, and those proteins can turn out to be a functional one. So one of the example that I would like to show you is, I guess, CD44 protein variants. I'm not sure. Yeah. So these, this is one of the gene, right? And CD44, it has these many exons. You can see they are numbered here, right? 5, 6, 7, 8, 9 to up to almost 90. The main protein is like this. That means it will have these many number of exons. We are not talking of the protein. Obviously, the proteins will have those many respective amino acids. We don't know in one exon how many amino acids can be formed. So we are not talking about the protein as of now, but obviously the DNA or the gene will translate to the respective protein. So if you look at these variants, let's say for example CD44E, it has the first part intact, but it does not have this part at all. So it directly starts from C12, C13 and C14. So these exons are there, rest of the exons are not there, and the last part is intact. So this is another variant of protein. Similarly, there is CD44V310. That is one variant where probably first 2 or 3 blue exons, they are missing. So this is the result of RNA splicing. That means respective exons are being removed, and these proteins, they have certain functions. Some of them can be inactive. It is not that always the RNA splice product will be active. In some cases, this splicing can result into inactive protein, but in some cases that is deliberately done to have a certain different function. So that is just something that I wanted to show you here. So, and that is what is the interrupted genes, right? Or split genes, which is the result of the RNA or different RNA splicing. RNA splicing does happen commonly within a cell, but depending on which exons are removed, that RNA splicing can lead to different variant of the protein. And those particular genes, they'll be called as interrupted or split genes. And this is simply to the fact that there is exon and intron present in the particular DNA segment. Any questions here? Anyone? Okay. Then the next type is the overlapping genes, or what is also called as there is another type called alternative genes, not the common names. So when a single sequence of DNA codes for more than one protein, right? Or there the alternative genes which are found when exons from different discontinuous genes, they get connected, forming several new combinations. So again, this is something similar to your RNA splicing. So the alternative genes, these are found when exons from different discontinuous genes. The main word here is different discontinuous genes. They get connected, forming several new combinations, right? And again, this does not happen so commonly within the cells, because these are specialized mechanism for certain specialized proteins to be synthesized within the body. So that is why these alternative genes or overlapping genes, they come into picture. And this was identified by this fellow called Gilbert. And the hypothesis of formation of different protein, that's what was given as Gilbert hypothesis. So that is the overlapping and alternative genes. Similarly, there is something called as transposons, which are known as jumping genes. So the DNA segments from one position can move into another position in a genome of a cell. And this is again going back to the fact that during the unwinding and winding of the DNA, a particular stretch of DNA is broken, and in the repair process, that segment is put somewhere else by mistake, and that is called as jumping genes or transposons. Again, this can lead to either malformation in the cell, or it can lead to the regular course of action, depending on what segment of the gene, where is it attached, and all of those factors. If it is within the intron, there shouldn't be any problem, or if it is within the non-coding sequence, again it shouldn't be a problem. But if it is in between certain gene sequence, obviously both the genes, they may not be functional. So that is something like transposons or jumping genes. And the last type, that is the gene families. Again, we have seen these gene families. When the multiple copies of the gene in eukaryotes, they are called as gene family. Like for example, skin, we had seen there are multiple copies. Skin color, I mean, not for skin. Even for hair color, there were multiple genes. So whole of that category of gene is called as one gene family. Or sometimes there are similar functioning genes which are present together. They are also called as gene family. It's just group of genes or multiple copies of one particular gene. That is also a gene family. And pseudogenes, this is a DNA sequence that resembles a gene but has been mutated into inactive form, right, over the course of evolution. So what happens is there are several genes which has a similar sequence to the normal gene, but due to the over the period of evolution, they have been mutated into inactive form. That means they are no more functional. Hopefully, inactive means they are no more functional. And again, and that is that particular gene segment is called as pseudogene, which is just the which is actual gene, and it can be mutation at one specific nucleotide, or it can be a particular segment of nucleotide, depend on what that mutation is, or there can be multiple single point mutation. So these are the different types of the genes that we know of. Any question here? Anyone? Okay. So then the second last topic of this chapter, that is Human Genome Project. So again, the name itself says quite a lot. This is the trying and identifying all the nucleotide bases within the human genome present in one particular cell, and also identifying their function. So this was the comprehensive international research effort to map entire human genome. So within one particular cell, whatever 23 chromosomes, rather 24, including this X and Y, those were present. So determining the sequence of nucleotide in those chromosomal structure or DNA structures, and to study the functions of each of these human genes present in the chromosome. That is the Human Genome Project. It is also called as International Human Genome Sequencing Consortium, because it was like combination of efforts from all over the world. I guess quite a few countries, they were involved in this, and just some data points. So total base pair, there are approximately 3 into 10 raised to 9 base pairs, and the expense that was expected was 9 billion US dollars. And again, a very fancy example of how much longer the human genome is. So if we type that each nucleotide in a book, 1000 letters per pages, so one book can have 1000 pages. So 1000 into 1000, that will make one book, and such 3300 books you will need to type out whole of the human genome. So that is like the length, you can imagine. It started somewhere in 1990 by this fellow James Watson, again from the Watson and Crick duo, as its first director of the Human Genome Project. And the first draft of it was ready by June 2000, but finally eventually it was completed in April 2003. That's when the whole of the genome was known. But still, whenever we say this, in April 2003 the Human Genome Project was completed, it does not mean that everything is known from the genome. There are still quite a few things which we are not aware of, especially in terms of functions of quite a few genes. We know the sequence, but we don't know what exactly is their function, or there are many genes which are involved in 2 or 3 different functions. So we need to know all of them. So that functional aspect of this genome project is still going on. Sequence wise, we know almost everything. So that is just few basic aspect of the Human Genome Project. Now, what were the goals? Why was it done, this Human Genome Project? So obviously, to sequence the entire human genome, storage to store the database of this nucleotide sequence, and so that it can be accessible to all the researchers all over the world. So whosoever is working on a particular gene or so, they can access this database and they can identify a particular gene. So what used to happen before Human Genome Project is something like, let's say if I'm working on a particular gene, and if I want to know the sequence, I had no other source rather than going through the whole process by myself. And similarly, if one of the other institute also working on the similar gene, they will also do the same process again and again over. So if there are 10 people working on one particular gene, those 10 people used to do it simultaneously by themselves. whole of the process of this, whatever this human genome project involved. So to avoid that, it was waste of effort, waste of money, and waste of time as well. So to create a database, so that if I want a particular gene, I will just go to the database, take the sequence, and do whatever I experiments that I'm supposed to do. So that is, the storage was one of the very important part of the goals. Identification, to identify all those genes, which are almost 20 to 25,000 genes in the human DNA. Miss, earlier, before it started, it was known approximately. Now we know there are almost 30,000 or so genes. Physical map of the genome. So to obtain the physical map of the human genome by cloning the DNA into what we call as YACs, that is yeast artificial chromosomes, or BACs, that is bacterial artificial chromosomes, and cosmids. Cosmids are again specially designed vectors to clone the DNA fragment into these vectors. So these physical map of genome is very essential to know which gene is located at which position on which chromosome. So that is what is physical map of the genome. So that gives us quite a good idea. So it's obviously like if it is chromosome 1 or 2 or X or Y, it will be always good to know particular position, because the interaction of these genes will be different, or during gamete formation or during meiosis process they might have a different role, and so on. So there are various aspects to this physical location of the gene on the genome. So that map of the genome was like required to be known by them. Genetic markers, again this is to identify the thousands of genetic marker and map them into the genome. Genetic markers are like like let's say if we are traveling from one place to the other, there are certain specific landmarks, what we call it, right? We identify them and then we say, okay, we are going on right track. Something like that happens in the genome as well. So there are specific nucleotide sequences present in the whole genome or whole chromosome, and they can be identified as genome markers. So let's say if I'm working on a particular segment of chromosome, and then I can use those genetic marker to sequence a particular gene. So that is the function of this genetic markers. Technology, one of the most important part of this whole of the human genome project was to design or develop new methods or new technologies, especially related to the genetics, such as gene cloning sequence contigs. Contigs means continuous gene cloning sequences, or sequencing of the complete genome. So the techniques were required for high throughput sequencing of the genes. High throughput means very fast and very efficient methods of the DNA sequencing. So those technologies, they needed to be developed by them, which exists nowadays because they are very efficient and they are still being used very well. To transfer the related technology, and not just to develop but also to transfer these technologies to the various industries or different researchers all over the world, so that they can also use it. That was one of the goal of this human genome project. And also one of the major one of the important goal is ELS, that is ethical, legal and social implications of the results that will be obtained from this project. So to tackle those issues, that was also one of the goal involved in the human genome project, specifically like because there are certain ethical issues when it comes to studying the any living organism. So that's when these issues they were raised, and they had designed certain what we say ethical guidelines to these studies, and that was part of this whole project. So that was, these were all the goals of the Human Genome Project. Anyone has any question? Pratham, I hope this part is clear.
Yes.
Now, what was observed at the end of Human Genome Project, or the salient features of the human genome? So there were quite a few parameters, and these are more factual. So what we know as of now is that human genome contains more than 3 billion nucleotides, and to be specific, it is actually the statement is wrong here. It contains 3.1647 billion nucleotides. That is what is known. There are 0.1% of plus minus. 0.1% of the billion can still go to almost a million or so, but that is a difference between different individuals, right? And that can be of different nature. But the major chunk of the human genome has been identified in all over the world. So that is like these many billion nucleotides in a particular cell. An average gene is formed of the 3000 base pair. Average gene, that means the length of the gene was 3000 base pair or 3000 nucleotide long. And the largest known gene has like 2.4 million base pairs, and that is dystrophin. It is one of the protein which is required for building the cell cytoskeleton. So that actin and myosin, all that, or during the cell division there is quite a lot of cell cytoskeletal movements, or during the cell migration from one place to the other, these proteins are required. So that is one of them is dystrophin, present in the cell cytoskeleton, and it is one of the largest known genes so far with 2.4 million base pairs. Total number of functional genes, like I mentioned earlier, it is 30,000. Means before the Human Genome Project was even started, it was thought that there will be somewhere around 50,000 to 1 lakh genes. But later on, when the Human Genome Project was completed, it was identified that there are almost 30,000 genes within the human genome. And the, like I mentioned this fact also earlier, the function of more than 50% of genes are not known yet. We know the sequence, but we don't know what they are coding for, or what they are, means obviously protein sequence can also be predicted, but their exact function, what is that protein doing in a particular cell, that is not completely understood yet. So there are almost 50% of the genes, and again this data will keep changing over the time. It might have gone to 60% known and 40% unknown as of now, but that is, there is still quite a lot that we are yet to do. Less than 2% of the genome sequence code for proteins. So out of this total genome sequence, that is 3.1647 billion nucleotides, only 2% will code for proteins, and the rest of 98% is the non-coding part of the genome, which,
So,
yes.
so but if 98% is non-coding, that means that they don't have a function, right? So you can say that their function is known.
No, they do have architectural function in terms of building the particular DNA fragment around the histones and so on. They do have certain functions. Plus, there are promoter sequences, there are these satellite DNAs, there are telomere sequences, all of these, right? There are rRNA sequences. So there are quite a lot of these, which does not directly translate to proteins, but they have enormous amount of function within the genome.
So, but there'll be like a very tiny amount compared to how much the protein genes are there, right? Like
Mm,
all the rest.
Not
Like,
necessarily, Pratham, because when we talk of protein, one mRNA molecule can give rise to multiple copies of protein. That's what we had seen, right? But at the same time, one particular mRNA, sorry, not mRNA, rRNA stretch will always give rise to one rRNA copy. So, sorry, one DNA stretch, which is coding for rRNA, it will be 1:1, right?
Yes.
So there, if I need 100 copies of rRNA molecule, that means I need to have 100 stretches within the genome. And that way, and you can obviously imagine the rRNA is required in tremendous amount because the ribosomes, they are required every now and then for every protein that is being synthesized in the body. Plus,
So, but like similar to proteins, can't it like one stretch give multiple copies?
correct. But then if you differentiate in terms of the amount to the number of times it is required, this is drastically different in terms of protein to RNA. And also, like I said, cell division keeps on happening. So those telomere sequences, they are also like non-coding part. Or this poly-A tail, no, not the poly-A tail, these what we call the promoter sequences or the regulatory switches within the DNA fragment. So there is quite a lot. There is something called as within the histone binding itself. Histone always binds to a certain segment of DNA, right? And that is when it acts as that switching it on and off. So those segments, all of these, they are present everywhere. You can imagine the number of nucleosomes present. So the number of switches or these what we call, there is specific term to this, I can't recollect, but yeah. So all these sequences, they actually amount to quite a lot of genome sequence. And of course, like you said, there is some DNA sequence which may not have any function, but that is simply required there to let the DNA structure in certain folding arrangement. That is just the architectural requirement of the DNA sequence. But
So,
yes,
the things that code for stuff other than proteins, like mostly that'll be RNA. That's more than the protein part, like the amount of nucleotides that are there.
yeah.
So, like, and like if you see the figures, that should be like that should be a bit around 48%.
No, sorry. Where?
So, like, if we know, like, let's just say we know 50%, and two of them are proteins.
No, this is not out of 50%. This is out of 100%,
Yes,
2%
sir.
genome sequence.
So, so like functions of 50% of the genes is known, right?
Correct.
Yeah, and so out of the total genes, only 2% make for proteins. So we also know for like if we so we also know 48% more.
No, no, we know all the genome sequences. We know all the genes within the, it just the functions are not known. Were you saying something different, or am I saying something different?
Oh, but then so the sequences are known,
Yes,
but the functions are not known.
functions are not known. Correct. We can also, from the DNA sequence, we can also know the predicted protein sequence, right? So maybe at the next class, we will do a little bit of exercise. I will show you how it is done, because that's like more practical aspect of whole of this. So
So, but this line less than 2% of genome sequence code for proteins. So, like then it doesn't really fit, right?
why not?
So, so less than 2%.
So I don't know if you are calculating the see the number of proteins we can count. Probably, let's say there are 30,000 genes, so there will be 30,000 proteins, right? But if you calculate the total number of introns, total number of histone binding molecules, total number of telomeres, total number of rRNA molecules or tRNA molecules, and all of these which are present in the DNA sequence or the genome sequence, right? If you make total of this, it will be far beyond 30,000, right? And that is when that 90% of the genome is beyond all of these numbers, which is like coding, which is like for the non-coding part of the proteins. Means that is not coding for the proteins. So that is that remaining 90%. And only 2% which is constituting in these 30,000 genes.
So like the statement less than 2% of genome sequence code for proteins is known. So does that mean the code, or like does that mean the sequence, or are they implying the function?
Only the exon sequences.
So they are implying the sequence and not the function.
Yeah.
So they are saying that only like if there are like 100% proteins, they only know the sequence of 2%.
No.
Or do they only know the function of 2%?
No, it's not related to function at all. So I have, let's say I have 100 base pairs, right? Yes. Out of these 100 base pairs, only 2 base pairs, they are coding for the protein. Now this analogy is really weird, but let's say I have 1 lakh base pair.
Okay, yeah.
Only a part of it, which is 2%, which I guess 2,000. Yeah, 1 lakh or 2% is, yeah, 2,000. Only these 2,000 base pairs they will code for protein, and the remaining 98,000 will be coding for these other functions of the genome.
So out of the other 98%, some part will be introns, like which don't code for anything, and some part will be the ones that don't code for proteins but code for something else.
Correct. That's
and out of that we know 50%.
Yes, now it's correct.
Okay. So 50% can be distributed among any three.
Yeah. Even within the non-coding sequence, we are yet to know the function of each of those non-coding sequences as well. They can't be so randomly present within a cell, right? Even we talk of a particular intron or particular sequence which is binding to histone or whatsoever, or even telomeres, we know quite a lot now. But like each of these sequence which is not coding for protein has a certain function as well, and we are again not aware of those functions completely, and that is also true.
So like, but earlier didn't we say that introns are only there for structural integrity, and like they don't actually do anything? So even if they are changed, nothing, like there'll be no effect on the body.
As of now, we know this fact, but that may not be the truth, right? Over the time, when we keep doing research, that's how we discover a few things, and probably that could be one of the fact, because that is when I said nothing in our body has been found so far which is just useless. Had certain function at least earlier, now it may or may not have function, but it surely has certain function in whichever time frame.
So isn't it just to counter mutations? So if the whole protein is filled of exons, there'll be a lot of mutations. But if there are a lot of introns also, then some mutations can be
within introns. Yeah.
So like,
but that's still a function.
Yeah, so but that we know, right? I mean, not like, sir, like a function as in, like a major function.
Correct. That is true. That can be the case. Okay, so I guess let me just finish this part and then we can stop. So that is, this less than 2% genome we saw. Repetitive sequences that do not code for protein make up to at least 50% of the genome. Repetitive sequence as in, there are quite a few stretches which are repeated several times. That makes, and this includes everything that is present there. Like I mentioned an example of rRNA molecule, which needs in many copies they can be called as repetitive sequences. Or even we had seen those earlier types of DNA segments like micro and mini, satellite, and so on. Those are also included in repetitive DNA sequences. They constitute 50% of the genome, and are stretches of DNA sequences. They are repeated up to 100 to 1000 times. So you can imagine if one DNA segment is repeated 100 to 1000 times, and if there are already 1000 copies of such repetitive sequences, then into 1000, so that is like quite a lot of nucleotides. Approximately 1 million copies of 5 to 8 base pairs, what we call as very micro satellite DNA. They are repeated sequences, and they are clustered around the centromeres and the telomere sequences, which represent the junk DNA. Now, junk DNA does not literally mean junk, which is non-useful, but obviously the centromere and telomere, they are very specific function in terms of cell division processes. So that's where this almost a million copies of these 5 to 8 base pairs, they are present, which is called as junk DNA. Genes associated with particular human diseases or disorders, they have been identified. For example, there are quite a few genetic disorders in terms of diabetes or in terms of metabolic disorders and so on. They have been identified through this human genome project, because once we know the function of particular gene or particular protein, we can elucidate if there is a mutation or if there is change. Obviously that gene is responsible for it, so that way it has been identified. Chromosome 1 is the largest human chromosome with the largest number of genes. That is almost 2968 genes in chromosome 1. And chromosome Y is the shortest chromosome with least number of genes, that is 231, that is 231 genes present in them. You can see almost 10 times difference between chromosome 1 to chromosome Y. By differential splicing, a gene may code for 2 or more protein. This is what I had shown you in that example of CD44. Right? Depending on which exons are going forward for after the splicing, they will code for 2 or more protein. About 1.4 million bases of DNA differences have been identified in human. So that is like almost 0.1% to 1%, I would say, or less than 1%. Single base differences, and these are specifically called as single nucleotide polymorphism, or that is short form as SNPs, or they can also be called as SNPs. So like if I do the genome sequencing of my own cells, and then if you do it for your own cells, there will be difference of almost 1.4 million base pairs. And that is what gives us the individuality. Yes?
So shouldn't it be much more? Like around 10 million?
Uh,
Uh, so because like before we said that there are like 3 billion genes, and the plus minus is 1%, right?
so
So that
Pratham,
should be
out
30
of
billion.
that 2%, which is coding for protein, 1.4 million is quite a lot.
Okay. So you take the two.
Because
Okay.
that's where the differences in subtle structures of the proteins and this will come into picture, and that is quite a huge amount, right? So there the proportion in terms of percentage is quite a lot. So that leads to the single nucleotide polymorphism, which is due to the single nucleotide change, and that if it leads to the amino acid in a particular protein, it can be useful in identifying locations of disease associated sequences and so on. So again, this goes back to the same fact. And the part of the DNA contains repeated DNA sequences, which we call as satellite. So these were like different features that were identified in the human genome project. If you have any question, so we will look at in the next class the strategy and methodology and there are, I guess, hardly 4 or 5 slides left. This part I will give you for self-study, because these points we have already discussed, like identification of the genome, identification of the sequence for diseases and so on, metabolic functions and so on. So if you read this application, you will see quite a lot of repetition of what we had discussed, so that you can self-study. And then the last process is the DNA fingerprinting, which we'll see tomorrow. And there the only techniques are more important, and that's it, and that will be the end of this chapter. So tomorrow we will surely finish this particular part.